Today, Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, and Dr. Christian Ross, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to development and validation of tests for SARS-CoV-2. Please remember that during this town hall, we are not able to respond to questions about specific submissions that might be under review. Now, I give you Toby.
Thanks, Irene. Thanks, everyone, for joining us again. I have a few updates of actions that we've taken recently. Last week, we authorized the Lucira Check-It COVID-19 test for over-the-counter use for single-use at-home screening tests. And this one was previously authorized for prescription home testing. We also authorized the Amplitude solution with the TaqPath high-throughput combo kit from Thermo Fisher, and that is a test that has the ability to perform up to 8,000 reactions within a 24-hour period. And then we also reissued the SalivaDirect test from Yale for serial screening with self-collected saliva samples at home. So those were a few notable authorizations from last week. And then we also had a question last week that stumped us a little bit about the difference in terminology that we use in the email updates, the CDRH new email updates that our comms office prepares to send out updates to the listserv about website updates. And those include new or newly posted EUAs. So we did check into that, and I can clarify that when the email says that it is a new posting, that it is a new test that did not have any previous EUAs and where we issued a letter of authorization. If it says that it is a reissue, that means that we reissued the letter of authorization, and that's generally used for updates that change any of the details in the letter of authorization, including updating the intended use, the conditions of authorization, et cetera. And then revised generally means that we issued what we refer to as a granting letter. So those are the letters that you'll see if you expand the little plus sign on the EUA list, and there'll be letters that we grant to update some details about the EUA. And those are generally used for updates that require FDA concurrence, such as changes to labeling, but they don't impact the letter of authorization. And then if the email says updated, then that is referring to updates to labeling, where we might have posted new labeling without issuing any letter. So without reissuing the letter of authorization and without issuing a granting letter. And that's usually used for minor changes, to the labeling, fixing typos, minor clarifications, things that are generally not substantive to the performance of the test or don't, don't involve evaluation of new data. And then we also use that updated labeling when we are adding results from the reference panel to the labeling of the test. Okay, so those are my updates from last week. And now I can go through the questions that we received ahead of time. So one of the questions is asking whether a test developer needs to notify FDA if they want to take their EUA off the market, and asking whether they would be removed from the EUA page. So we do ask the test developer inform FDA if they intend to take their EUA off the market. You can do that just by sending in an email to the CDRH EUA templates mailbox, and we will respond to you and go through the process with you that way. Generally, if you're planning to take a test off the market, we will revoke the EUA and the revocation will note that it was per your request. And we would remove the test developer from the EUA authorization list on our website. Similarly, if a developer has notified FDA through the policy in our guidance, we would also ask that you let us know through an email to the templates box if you plan to stop marketing that test. And then we would generally remove you from the notification list and include, you can see on the removed list, it includes an asterisk if they're removed voluntarily. Let's see. Next, we have a question about the process and timing to discontinue EUAs and require 510Ks or de novos. And we have discussed this a little bit previously on calls. And we've mentioned before that I can't comment on a timeline specifically, but we are working on a transition plan for devices that are offered under EUA. And this is included as a guidance document on the center's guidance priority list for FY21. If you take a look there, you'll see the transition plan for medical devices distributed under enforcement policies or emergency use authorization during the COVID-19 public health emergency. Additionally, it's important to recognize that generally, unless they're revoked, an EUA is in effect until the public health emergency is terminated. And the termination of a public health emergency does not typically happen on a particularly accelerated timeline. You can see that there are previous public health emergencies like Zika and Ebola that still have not been terminated. Now, we have authorized the first test for marketing beyond the public health emergency with the Biofire de novo, and that does open up the 510K pathway for other molecular diagnostic SARS-CoV-2 tests, both individual or single analyte SARS-CoV-2 tests, as well as multi-analyte panels, which the Biofire test is. But that de novo does not impact any other EUAs other than the Biofire EUA for that same identical test, which was revoked concurrent to granting the de novo. Biofire's other EUAs and other developers' EUAs were not impacted. So we can't anticipate when the public health emergency would end, but we are working on that transition plan, and we are committed to ensuring that the public has access to a wide variety of test options for COVID-19. And we have no intention at the moment of stopping review for EUAs or revoking based solely on timeline or anything like that. Our next question is about the home collection template and the requirement to establish a process with specific labs to identify the process of shipping the collection kit to the customer, and then the specimen back to the lab, and getting the results back to the individuals, and asking about whether we would accept an EUA request with a general description of the process with specific logistics being established by each lab, or whether the collection kit manufacturer needs to establish those partnerships with specific labs. And so generally we do allow the submission of, or we would consider a submission of an EUA request for a collection kit, a home collection kit, that with the intent of it being used by multiple lab partners. We would recommend that you develop and submit an accessioning protocol that includes information about how long the shipping stability supports the sample stability, rather, and the procedures that a lab should perform before accepting a sample for testing. We would want to see that you've built in assurances that all the labs would use the same home collection kit instructions with minor edits for brands that we do through labeling review, and that there would be assurances for checking that purchasers of the product are using the appropriate assay supported by the collection kit. And we are happy to have further discussions in the context of your particular submission on those details. We have a question about at-home tests and whether they can be performed in a public setting, like a college, a school, or workplace, as opposed to strictly in a home. And if so, are they exempted from CLIA regulations, even if they're performed by the facility, not the individual? So this one is really a question for CLIA. We would recommend that you reach out to CLIA for a definitive answer. But our understanding is that an at-home test can be used for self-testing in a non-home setting. But if the test is performed by someone else at the facility, then the facility would need to have a CLIA certificate, which would generally be a certificate of waiver in that situation. We had a question following up on a call, or on a question we addressed last week about the validation of multianalyte molecular tests and the use of archived rather than contrived samples. This question is asking specifically about validation of multianalyte antigen tests, and in cases where the sample type used by the antigen test does not survive the archival process. So the question is asking whether multianalyte antigen tests can leverage contrived samples for the initial authorization with a post-authorization commitment. And generally, we would not consider it to be acceptable to use contrived samples for initial authorization of a multianalyte test. We would want to see a prospective study to support the EUA request that includes an FDA-authorized RT-PCR for the comparator for SARS-CoV-2 and an FDA-cleared comparator for all the other claimed analytes. And if there are technological considerations that preclude the use of archived specimens, such as what was mentioned in this question of the sample type not surviving the archival process, we would encourage you to submit a pre-EUA to discuss the next steps for your specific situation. We have a question about pre-submissions and if there's any indication on review priority for SARS-CoV-2 pre-submissions or QSUBs. We have obviously seen delays in CDRH on pre-submission and 510K reviews during the pandemic. So this question is asking whether that applies if a pre-sub is submitted for a SARS-CoV-2 assay that does not currently have an EUA authorization. So generally, we are broadly committed to ensuring that the public has access to a wide variety of test options for COVID-19, and we do intend to continue to review SARS-CoV-2 pre-submissions to address public health needs. So to some extent, so COVID-19 related pre-submissions are one of the categories of pre-submissions that we do continue to review, and the review of those would be prioritized based on the public health needs. So we would still consider the particulars about the device in question as to whether it would be a priority review. Our next question is about rapid antigen tests based on lateral flow devices and whether we continue to review submissions for professional use in point of care with settings with a CLIA certificate of waiver, or whether we would view that as low priority unless the submission has data for self-testing in a home use setting. So we do have many factors that go into our prioritization decisions, and unfortunately not all of those factors are able to be made publicly evident since they do often consider other submissions that are in-house. But we have previously stated that we're prioritizing review of EUA requests for tests that increase testing accessibility, which includes point of care, home collection, at-home tests, for example, or that would significantly increase the testing capacity by reducing reliance on test supplies or tests that have high throughput and are widely distributed to address public health needs. In this case, if it is a test that is designed for home use, we would generally want to see that it is validated for home use. We are still reviewing point of care tests as well, but depending on how the test is designed and what it is intended for, we would want to have further discussions about that. So if you have questions about a decision about your test in particular, please reach out. Our next question is about a clinical trial designed to enrich for asymptomatic positives, asking for, this is for a nucleic acid test with a lateral flow strip readout, and asking whether it is acceptable to recruit asymptomatic subjects with recent positive test results if an equal number of asymptomatic subjects with recent negative test results are also recruited. And the question additionally details that due to the workflow where the subject is guided through result interpretations with an app, the individual would not know which they would not know the which readout is positive or negative before inputting it into the app. So their knowledge of their prior test should not create bias, according to the interpretation of the individual asking this question. So generally, there are a variety of ways to enrich your clinical study. The proposal in this question generally appears acceptable. We would want to make sure that you're using the comparator method as the result that you're using to enrich your clinical study population. And if you have further questions or want to talk through the design, you can reach out with a pre-EUA to discuss that study design as well. Chris, did you want to add anything about that one, about the study design, or just to come in with a pre-EUA?
Yeah, I think a pre-EUA probably is the right method because there are, I think nuances to this approach. You could also consider, other patient factors, risk factors, where folks may be exposed to kind of further enrich population. So there's a number of ways to do this.
Great, thanks, Chris. All right, the next question is about multi-analyte RT-PCR tests, specifically SARS-CoV-2 and influenza A/B, and whether we would find it acceptable to have an asymptomatic claim for SARS-CoV-2 in a case where software associated with the test is capable of reporting only a SARS-CoV-2 result if only that analyte is ordered. The question notes that there appear not to be any multi-analyte EUAs that have an asymptomatic claim. And so they're asking about that. The questioner is correct that we do not currently have any multi-analyte EUAs that have an asymptomatic claim. At this point, we don't have sufficient data supporting device performance or a public health need for an asymptomatic influenza screening claim. For SARS-CoV-2, we obviously know that there's many reports of asymptomatic infection and numerous studies that show detectable viral load in asymptomatic populations. So we have obviously granted asymptomatic screening claims for many SARS-CoV-2 tests at this point. So a test that's capable of reporting either a SARS-CoV-2 result alone or the multiplex result, depending on the patient population that's being tested or the individual's circumstances, may be appropriate. We would want to see that there are sufficient controls to ensure that the user has the tools to report the appropriate results to the appropriate individual. So we would recommend that you submit a proposed plan, and then we can discuss that further, including, what the labeling would look like and how the workflow would be handled there. Chris, anything you want to add on that one?
No, that's perfect. Thanks.
Great. All right. Let's see. Our next one is another one about respiratory multiplex panel. This one is about transitioning from an EUA to a 510K and the inclusion of multiple RT-PCR instruments. The question specifically is asking whether the prospective clinical study for the 510K could be conducted on the instrument with the worst limit of detection. This is something where we would generally want to have some further discussions about your approach. The proposal that you laid out might be acceptable. We would recommend providing data establishing that performance, low analyte levels, precision and reproducibility are similar between the instruments. And we would recommend that when you're pursuing the 510K pathway, that you submit a pre-submission to discuss your proposed validation strategies and your comparator method for review. Right now, there's quite a bit of information in previous 510K decision summaries for flu and RSV, and we hope to get the decision summary for the Biofire de novo posted fairly quickly. And so since the studies that establish performance for flu are likely to be similar to what we recommend for SARS-CoV-2, we would suggest that you take a look at those flu decision summaries to get a sense of what we would be looking for there. And then since we do have limited resource capacity for pre-submissions right now, we recommend that you maximize the efficiency of that interaction by including specific questions that are targeted to your test and the remaining questions that you have after reviewing the publicly available information. Anything to add on that one, Chris?
No, I think there are instances in the 510K pathway where we've done this, and so I think you can find information there. We've also have the migration pathway, so I think that's got some good information. The migration guidance has some good information about comparing two systems.
Great, thanks. And then the last question that we have is about serology tests and the template. They're asking about the language in the template that's asking for a plan to assess the impact on new variants. And specifically, they're asking if we need to develop a test protocol and perform testing on new variants, and to clarify the output required for that section of the EUA template. So the template does state that for any viral mutations and variants that are identified as prevalent and or clinically significant, you should assess whether the resulting predicted amino acid changes in the viral proteins are critical to your test design. And if so, we would ask that those mutations and variants should be evaluated using clinical specimens or contrived, depending on availability, to assess the impact of the mutation or variant on your test performance. And then specifically, the template suggests that you provide your plan for monitoring for new and emerging SARS-CoV-2 viral mutations and variants on an ongoing basis, and for assessing the impact of mutations and variants that have been identified as prevalent and or clinically significant on the performance of your assay over time. And this includes providing your plan that includes information on the potential impact of the mutations and variants on your test performance, or explaining how the risk associated with the unknown performance of your device in samples from individuals with the variant can be adequately mitigated. So generally, a protocol that indicates how you would do that testing, along with the other information requested in the template, would serve as that plan at the time of your EUA request. And as we do that review, we would discuss that protocol and plan with you as needed. And that is all of the prepared questions and answers that we have, so we can turn it over to the live portion.
Thank you. If you would like to ask a question over the phone, please press star, then 1. Record your name clearly when prompted. If you need to withdraw your question, you may do so by pressing star, then 2. One moment as we wait for our first question. And our first question comes from Annie Wright. Your line is now open.
Hello. Last week we had a question about using clinical sites in the UK for the SARS-CoV-2 studies. So we were wondering if we had a clinical site in Canada along with other clinical sites in the US, would that be acceptable to the agency?
We do accept data from outside of the U.S. With the exception of generally for point of care and at home validation studies. We would want those to be done in the U.S. due to variations that we have seen, particularly in those areas. And then, Chris, do you want to touch on this one at all?
This would be a point of care study.
Okay, yeah, we would also want to see that done in the U.S. Yeah.
Yeah, I mean, if we did have data in the U.S. that also included a site in Canada?
I think that would probably depend on the volume of data that you have from the U.S. I think we would want to see the data specified in our template from the U.S. If you have supplemental data from Canada, you can certainly submit that as well.
Okay, but the predominant, basically, as long as you meet the minimum criteria for the U.S. For the template, specified in the template, conducted in the U.S., then that would be acceptable. If we include additional in Canada, but
right. I think so. Chris, do you want to add anything on that?
Yeah, I mean that's a safe bet, right? I mean, if you have international data from POC sites, then we start perhaps asking questions about, well, what exactly is the standard of practice at these POC sites? So it potentially, you could make that argument that it's similar to the U.S., but if you can avoid having to make that argument, that's just kind of one less thing that you have to justify to us.
Okay. All right. Thank you so much.
Thank you. And our next question comes from Elizabeth Brunelli. Your line is now open.
And this question is about home collection kit EUA submissions. Is it important to include product branding information in the EUA submission, or can that be submitted post-EUA?
So, that may depend on exactly what you mean by product branding information, but generally we do review the labeling, and so that would probably include the product branding that you're referring to. We do usually include in the EUA a condition about adding labeling where, for distributors that might distribute under a different name, and that's something that can be added after the initial authorization if you're looking to partner with different distributors. But the test name is included in the authorization, and so the name obviously would go on the labeling that we would review and authorize as part of the EUA.
So what we're speaking specifically to is that we have kind of a generic name for our home test, our home collection kit at this point, but we might change the brand name of it to include some branding that we've used previously for our laboratory.
Okay, so you could submit it with the fairly generic name if that's what you're planning to use right now, and then if you decide to update it, you would submit that update as a supplemental EUA request, and we would have to review and authorize that alternate labeling.
Okay, perfect. Thank you.
Yep.
Thank you. And our next question comes from Anna Powell. Your line is now open.
Hi, my question is regarding developing a home use serology antibody test. Is it required to report? Does it have a reporting requirement like some of the antigen home use test? Second part is, should we include vaccinated people in the performance study?
Okay, so this one is a little bit tricky. We don't have recommendations that we've put out yet for home use serology antibody tests. Generally, if there is a healthcare provider involved, which we would, all of the serology tests that we have authorized so far are prescription use, we would, the coordination with the healthcare provider can be how the test results get reported. Otherwise, we would discuss that further, and we would suggest a pre-EUA to discuss what we would want to see in that home use antibody test. And similarly with your question about including vaccinated people, that is an area that is emerging now, obviously, as more and more people are becoming vaccinated. And so we would suggest that you submit a pre-EUA with your proposal, both for reporting and for inclusion in your validation study, so that we can discuss specific to your approach.
So what is the response time if we submit the pre-EUA to address the basics of protocol and the questions like this for a home use serology test? We heard that there's a very low priority for this particular test. Is that true?
We would evaluate the priority of the request when it comes in considering the public health needs at the time, as well as the other submissions that we have in-house. So I'm not able to give a specific timeline there.
Okay, thank you.
You're welcome.
Thank you. As a reminder, to ask a question, please press star 1. Our next question comes from Stacy Hausman. Your line is now open.
Hi, thank you for taking my call. I have a question related to the molecular point of care template. Specifically, it says if we do a prospective enrollment, looking for 30 positives in the symptomatic population. And after a period of 2 weeks, the template states that we can move to using bank samples. And so what I'm wondering is, is the 2 weeks, is the 2 week time period just 10 days, or is it wanting us to prospectively enroll for a period of 14 days? Just checking, because the business days, if we only enroll on business days, then that would be closer to a 3 week time period, and we just want to make sure that we're following that appropriately.
Sure. Chris, do you want to address this one?
Yeah. Okay. Yeah. I mean, 2 weeks is 14 days. So I mean that's the kind of just taking it at face value.
Okay.
Really the point there is you've made some kind of good faith effort to collect some prospective samples to at least have that portion of it be kind of an all-comers type of study. I mean, that's very, very valuable information for us. So I would say, just go with the 14 days.
Okay. All right. 14 days of enrollment. Yeah. Okay. Thank you.
As a reminder, to ask a question, please press star, then 1, and record your name clearly when prompted. Our next question comes from Elaine Allen. Your line is now open.
Yeah, hi. Thank you for this opportunity. Toby, did I hear correctly that there may be some information on the website somewhere regarding the transition plan from EUA to 510K? Is there some general information out there?
I did mention something being on our website. It might be overstating it to call it information. The name of the guidance document is on our guidance priority list.
Guidance priority list. Okay, so I'm not sure where that is, but I'll try to find it.
If you Google, I think FDA guidance AB list, it should come up pretty close to the top.
Guidance AB list?
Yeah, we break the priority list down into an A list and a B list. And so we often refer to it as the AB list. And I know that when I have Googled, guidance A space B list, it comes up.
Oh. And in regulatory 20 years, that's the first time I've heard of that one for FDA. So thank you very much.
No problem.
Thank you. Our next question, the first name is Ella. Your line is now open.
Hi, I have a question related to what was asked previously on the point of care and at-home test. So I understand the usability study for sure has to be done in the U.S. What about the clinical study with the 30 positive and negatives? Is that okay to be done outside of the U.S.?
Since the setting does impact the clinical for those tests, we do want to see that they are done either in the U.S. or that you're able to provide justification demonstrating that the outside of the U.S. sites are representative of the U.S. sites where those would be done. So we would want to see that the operators are representative of operators in a U.S. CLIA waived setting, for example, for a point of care test.
Okay, thank you. But the clinical evaluation is not done by the operator. It's done in a lab, correctly, with clinical samples.
The clinical evaluation should be done in the setting in which the test should be used. Chris,
okay,
do you want to
thank you for
weigh in on that?
Yeah, I mean, an important aspect of performance is having a test that the user can, perform correctly, right? So, I mean, if you've got a POC claim, I think ideally we're going to ask for POC users to be running those samples.
Yes, but from the guidance, I understand specifically for POC, it is recommended to have 5 to 6 trained operators in 1 to 2 different CLIA waived labs. So I'm just trying to understand the 30 positives and 30 negatives. How do they interact with those samples taking done in the CLIA waived setting?
So are you talking about bringing samples? You're talking about testing banked samples, for instance, from internationally collected bank samples?
No, so my understanding was that for, let's say, a high complexity lab, you would only need the clinical evaluation of 30, minimum of 30 positives and 30 negatives. And then for a point of care claim, you'd in addition need the CLIA waived operators doing more tests.
Sure.
And so the 30 positives, 30 negatives, those should also be done in the US, or could they be done abroad?
I think ideally all testing, validation testing for a POC test, we would prefer it done in the U.S. Again, like we mentioned before, if it's done in an international site, then the burden is on you to establish that the procedures and practices used at that site are similar or identical to what would be done in the U.S.
Okay, thank you for clarifying.
Thank you. As a reminder, to ask a question, please press star one. One moment as we wait for any additional questions.
And I think while we're waiting on that, I just want to clarify for the point of care question. I think there may be some confusion about how to perform that study and about the language in the template when the template is referring to including those 5 to 6 minimally trained operators. Chris, that is for the 30 and 30 study. Is that correct?
I think our opinion is that that POC test is going to be run by folks that are minimally trained, and therefore we want to have the validation performed in a similar manner.
Yeah, thank you. Just wanted to clarify that for that caller.
As a reminder, to ask a question, please press star one. One moment as we wait for any additional questions. And our next question comes from Roxanne Chan. Your line is now open.
Right. Thank you. Yes. I have a question regarding the multi-analyte molecular test validation. So we're in the process of completing the validation work on a multi-analyte test that includes COVID and influenza. And we've been advised that it would be helpful if we put in a high-throughput platform. So we are considering doing that. But my question is that how extensive the validation has to be if we want to include now adding a high-throughput platform? Do we only need to do like adding an extra instrument type of work, or we will have to, repeat a whole set of validation, analytical and clinical?
Thanks. So, to clarify, the high throughput is really because that will help increase the capacity, the testing capacity, which is, what is needed at this stage for, as we try to get more routine testing done through routine testing programs. Chris, do you want to talk about the validation that we would look for there?
Sure. Yeah, I think, if the test is going to be distributed with a high throughput workflow, that's the workflow that should be validated. I think performance in a 96, just for instance, performance in a 96 well plate may be different than a 3 to 4 well plate, right? Heat transfer is different. Optical characteristics perhaps are different. So I guess what we don't want to see is, like a manual workflow done in the validation, and then kind of maybe just an LOD or study or something like that to validate the high throughput. I think what we want to see is that high throughput solution is kind of producing acceptable results, because that's really the solution that's going to impact testing the most.
So what you're suggesting is that it will be more than bridging studies then?
Chris?
Hello?
Oh, it looks like Chris is having some technical difficulties.
Yeah. Yeah. So what I'm trying to understand is, for instance, in the validation package, there will be, of course, the very first part is the LOD definition. And then after that, there will be a bunch of inclusivity, exclusivity, competition, co-infection, and clinical studies.
Yeah, so, I think that there are certain aspects of validation that would not be impacted. Mm-hmm. And obviously, we wouldn't expect to see those repeated. The portions that would be expected to be impacted, we would want to see, the validation with the high throughput platform or justification of why, what you're able to demonstrate is representative.
Okay.
Right. Unfortunately, it looks like Chris dropped the call. He got disconnected. But if you want more specific details, you can send that in to the mailbox, and we can try and get you more details on the technical side there.
Perfect. Very helpful. Thank you.
Yep.
As a reminder, to ask a question, please press star 1. One moment as we wait for any additional questions. Okay. Seeing that there are no additional questions, we'll turn the meeting back over to Ms. Irina here.
Thank you so much. This is